Welcome to the 2021 NDF Speaker Series on GNE Myopathy. My name is Angela Leck, and I'm pleased to introduce to you today Katherine Koczwara, who works with us at Yale as a grad student under the direction of Dr. Moko Leck. Today, she'll be talking about using patient cells and how they help us better understand GNEM and provide new ideas on developing and testing therapies. So you'll have the opportunity to ask questions in the chat box as she presents, and we will address them at the end. So without further ado, I'd like to turn over the screen to Katherine. Welcome, Katherine.
Hi, thanks, Angela, for that introduction, and also a huge thank you to the NDF for giving me the opportunity to speak today. So as Angela mentioned, I'm going to be talking to you today about some research that I'm doing in the Leck Lab involving some translational GNE myopathy research that I'm doing in cell models. So a brief overview of the structure of my talk is first I'm going to give some background into GNE myopathy that will be relevant to what I'll be discussing. Then I'm going to go over some of my research aims that I'm working on in the lab, namely developing some robust cell model systems of GNE myopathy, some reliable assays or disease biomarkers for the disease, as well as some work to further our understanding of the disease mechanism, followed by some concluding remarks and conclusions of the work that I'm doing. So to begin with some brief background into GNE myopathy, GNE myopathy is an autosomal recessive disorder that leads to progressive skeletal muscle atrophy and weakness in the legs and arms. It's caused by mutations in a gene called GNE, and as a rare disease affects about 1 to 9 in 1 million people worldwide. And characteristic of the disease is the presence of rim vacuoles and inclusion bodies in the muscle. So what you can see in this image in the right is in the patient with GNE myopathy, you see the presence of these rim vacuoles and inclusion bodies within the muscle fibers that you don't see in healthy individuals. So an important aspect of GNE is that it's an enzyme in the sialic acid biosynthesis pathway, and which is pictured here in this diagram on the right. And some important things that to take away from this diagram are that the GNE enzyme performs 2 steps in this pathway. So it has an epimerase function as well as a kinase function. So it performs 2 steps in this pathway. Second, that there's a negative feedback regulation that occurs. So the downstream process byproduct of this pathway, or CMP sialic acid, can actually go back and bind to GNE to regulate its activity. And lastly, and something that you should really take note of is that sialic acid is eventually used to sialate proteins on the cell surface. So it's added as a terminal sugar on sugar chains on the cell surface. And this is a really important thing to keep in mind throughout my talk, because this activity of sialic acid, its presence on the cell surface, is something that we can measure or look at or investigate to determine how well the sialic acid biosynthesis pathway is occurring, and by proxy GNE activity. Another thing looking at the genetics of GNE and the GNE gene itself is, as I mentioned in the previous slide, you can see it has 2 domains for its 2 enzymatic activities, the epimerase activity and the kinase activity. And the mutations that we see in patients with this disease are present in both of these domains. So you can see listed down here some of the very common mutations that we see in patients with GNE myopathy, and they're scattered throughout the protein. So to go into some of my specific research aims of the work that I'm doing in the Leck Lab, one of the first main goals that I have is to develop robust cell model systems. But to begin, I'm going to talk about some existing animal model systems and why we're looking to develop cell models as well. So there are have been several attempts and created GNE myopathy mouse model systems. So namely, the M712T mouse, as well as the D176V transgenic mouse. But unfortunately, there's some difficulties and challenges with working with these mice. So namely that a complete knockout of GNE in mice is embryonic lethal. So you can't get a mouse that is completely deficient of GNE. And secondly, the 2 mouse models, 2 of the mouse models that have been created This one dies of a renal disease at day 3, so it doesn't survive very long, and when it does, it doesn't develop the muscle phenotype that we're looking for. And this other mouse that is of interest had the muscle phenotype that we were looking for. However, it lost the phenotype upon successive breeding, so unfortunately can't use that mouse. So this is one factor is that there's been difficulty in creating a super reliable mouse model, but also there is some differences in the sialic acid biosynthesis pathway in humans versus non-human mammals. So namely the presence of this enzyme called CMAH. And what this CMAH enzyme does is it actually adds a hydroxyl group to sialic acid. So what this means effectively is that in non-human mammals, when looking at these sugar chains that are present on the cell surface, we can see that in non-human mammals, they have a mix of sialic acid as well as this hydroxylated sialic acid, where in humans, we only see the non-hydroxylated sialic acid. And this has actually been shown to have play a role in muscle pathology disease progression. So in this paper out of Paul Martin's lab, they were looking at what deleting this gene in a mouse model of Duchenne muscular dystrophy does, and it actually increased the disease severity. So this indicates that the presence or absence of this enzyme does play a role in disease severity of muscular dystrophies. So an additional reason for looking into human-derived cell models is that it does not have this enzyme that mouse and other non-human mammal model organisms might have. So in order to kind of overcome the challenges in working with non-human cell model systems, we wanted to establish a biobank of patient-derived cell lines. So a resource of cells that come from patients with GNE myopathy. And to do that, it required a huge effort and the generous donation from many patients with GNE myopathy by donating skin biopsies. So as you can see here, the way that this process works is there's several layers to the skin, the epidermis, the dermis, and the hypodermis. And we're interested in a cell type called fibroblasts that exist in the dermal layer of the skin. So a physician can take a very small skin punch or biopsy into the dermal layer of the skin. And this little skin biopsy or skin punch can be sent to the lab for us to extract the fibroblasts. And so the way this process works, it's a very complex process from receiving the skin biopsy all the way to extracting the fibroblasts. But I'm going to show you some of the steps that we can do in the lab to complete this process. So at first we received the samples at Yale. We received samples from 2 patient conferences, one that occurred in UCLA and one that occurred in Philadelphia. And these samples are brought back to lab, in which case we can sort of remove the adipose tissue or the layers of the biopsy that we don't want, and then process the tissue to expose the fibroblasts that are in the dermal layer that we're looking for. We can then digest the extracellular matrix, so getting rid of everything that really isn't the fibroblasts, then collect these cells at the bottom of a tube. So you can see highlighted in this tiny yellow circle is the fibroblast cells from the skin biopsy that we're looking for. And then we can place them in a flask to grow out in culture in our lab. And what this ends up looking at is if you look at these cells under the microscope, at early stages, you don't really see much, but then gradually cells start to grow out and proliferate. And finally, about a week later, you can see here was a tiny piece of that skin punch, and then all expanding from that are all of these fibroblast cells. So it's a really effective process in being able to extract these fibroblast cells from patients with GNE myopathy. So after we've done this and extracted some fibroblasts, the next step is to immortalize these fibroblasts so they can keep growing in the lab for a very long time. So we can use them and share them with other labs. And so the way that this works is we introduce an H-TERT agent and select the cells that successfully obtained this agent. We then confirm that it was integrated in a test called a PCR reaction. And then we can place the successfully extracted and immortalized cells in test tubes and store them in liquid nitrogen. So we have this biobank of cells in our lab right now of many vials of these various cell lines stored in liquid nitrogen. But given that GNE myopathy is a muscle-specific disease, so meaning it affects the muscles. We kind of want to take these cells, which are skin cells, and convert them into a more muscle-like cell, because that will give us the opportunity to study a more myogenic-like or more muscle-like cell type, which is relevant given that GNE is a muscle disease. And so the way that this works is we have this plasmid that has human myoD, so a transcription factor that is unique to myogenic cell lines, under control of a doxycycline-inducible promoter. And so we can take our fibroblast cell lines, transduce them with this construct that will convert them into a more myogenic-like lineage of myoblasts, and then we can provide different culture conditions to differentiate them into myotubes. And this myotube-like cell line is pretty much the closest that we can get in culture to muscle cells. And so looking at some images of the cells, one of the cell lines from the biobank, we can look at the fibroblasts compared to the myotubes of this cell line. And we can look at two factors that are very unique to muscle cells. So one is dystrophin, which shows up as green in these images, and the other is myosin heavy chain, which shows up in red in these images. And what you can see is that when we induce these cells to form myotubes, or these muscle cell-like type cells, we can see both in green and red these very long myotubes, similar to the myotube muscle fibers that we were seeing. So this is a really good indication that we're able to not only extract the fibroblasts from these skin biopsies, but also to convert them into a myogenic-like lineage. So currently where the status of the biobank currently stands is we've had the generous donation of 19 GNE myopathy patients, as well as 14 unaffected family members to serve as controls. And of these sort of 33 donations that we were given, we have the primary fibroblasts, the immortalized fibroblasts, as well as the myoD converted myoblasts, which is a total of 99 cell lines, which is an incredible resource that would not have been possible without NDF's generous support, as well as the donations of patients. And that being said, if any patients who have not yet donated to the biobank would like to do so, we are always on the lookout for more samples to receive. So feel free to reach out to the NDF to organize that as well. So in addition to the biobank of patient-derived cell lines, I've also been working to create a couple of GNE knockout cell lines. So these are cell lines that are completely deficient of GNE. So the first cell line that I created a GNE knockout in is a HAP1 cell line. So this is a cell line that just grows really quickly and is really easy to work with in the lab. And so what you can see here is I was looking to create insertions or deletions in the DNA of these cells in the GNE gene. So compared to the healthy sequence, I have two different cell lines, one that has an insertion of these two DNA bases and the other that has a deletion of these DNA bases. And this affects the expression of the gene. So you can see both at an RNA level, our GNE knockout cell line, we don't see RNA expression, which we do in the wild type, as well as our in-frame deletion band, and as well as on a protein level, we are sort of missing looking at the staining of the GNE protein. We don't see the GNE protein expressed in this knockout cell line compared to a healthy cell line. So in addition to that cell line, I also wanted to create a GNE knockout in a myogenic lineage. So have been working to create a GNE knockout in a muscle cell line we have in lab called MB135. And so the way this works is we kind of design the guides to create the knockout, use an RMP approach, so it's a CRISPR-based RMP knockout approach to knock out the cell lines, and then can analyze the editing and knockout efficiency. And so this is the step where we're currently at, where I have been screening a bunch of cell lines with a variety of different insertions or deletions. So basically changes to the GNE gene that should make it so there is no GNE expressed. And then I'm working to grow out some clonal cell lines, as well as perform some assays to characterize this knockout cell line. So in addition to the biobank, these two complete knockout cell lines are a very useful tool to have in the lab. And then the last kind of cell model system that I'm working to do is to utilize prime editing to correct pathogenic mutations. So what this means is I basically want to take the cell lines that we have in the biobank that have mutations in the GNE gene and correct them to a wild type so they no longer have the mutation and it's a way of correcting the cell from a GNE myopathy cell to a healthy cell line. And so a brief overview of how this process works is there is a prime editing protein, which is composed of a Cas9 nickase protein fused with a reverse transcriptase. And additionally, there is a guide RNA that has not only the guide RNA target sequence that will bind to the place in the GNE gene within the DNA, that also a template containing the sequence that we want to change the DNA to. So in our case, we put the healthy GNE sequence in this part of the RNA to then correct it from the mutation into the healthy cell line. And these two components form a complex within the cell. When we add them to the cell line, they complex, and then they can bind to the place in the DNA that we're looking to edit. And so in this case, this would be the place in the GNE gene that contains the pathogenic mutation that causes the disease. It will nick one strand of the DNA, and then it uses the template that we provided, the reverse transcriptase template, to synthesize new DNA. So this DNA wouldn't have the mutation in it anymore. And then this corrected or healthy DNA sequence strand can then be incorporated back into the DNA. And then through DNA repair, both of the strands could contain the desired edit. So basically, this whole process is working to, at the DNA level, so change the GNE sequence from the sequence containing the disease-causing mutation to a healthy sequence. And so what I've done so far in working on this is I've designed all of the constructs that I need for this, and first just began testing to see if I could perform this DNA editing at the place in the GNE gene where one of the common GNE mutations is, so at the M743T locus. So I first was just looking to create the mutation in wild type cells, so healthy cells at this locus, to show that I can in fact perform DNA editing at this locus. And so what you can see from these images are the DNA sequences, so the DNA bases of within the GNE gene that I'm looking to edit. And highlighted in blue is the place that I'm looking to actually edit the DNA. And what you can see in the cells that I didn't treat, we see that there is a T present. But then when I perform this prime editing, I'm able to convert about 11% of the cells to a C base. So this is showing that I was able to edit at this location within the GNE gene at an efficiency of about 11%. But this is in a HAP1 cell line, so not the cell line that's present in our biobank. So the next thing that I wanted to do was optimize this process, this prime editing process, for the fibroblasts, so the cells that we have in the biobank, because those are the cells that I'm eventually looking to correct. And to not get into the weeds of all of the optimization I did, which included like nucleofector settings, the solution the cells are kept in, the total DNA amount used, the ratio of the different plasmids or constructs I was using. But when all of the optimization was said and done, I have been able to see prime editing in a fibroblast cell line. So this isn't looking within the GNE gene, but it's looking to insert a CTT base in this region highlighted in blue here. And again, what you can see is that I was able to successfully edit the DNA, this time at a ratio of about 13% in fibroblast cells. And so moving on from sort of the results of these two experiments, the first one being editing the GNE gene in a HAP1 cell line, and the second set of experiments optimizing prime editing for fibroblasts, the kind of next step that I'm working towards is to actually use this technique to correct the pathogenic mutations in the GNE Myopathy Biobank fibroblasts. So in this case, I would have the fibroblasts from a GNE myopathy patient that have the mutation in the DNA that causes GNE. I would treat them with our prime editing constructs, and hopefully a subset of them would then have the mutation corrected, so corrected to a healthy GNE sequence. And then I can take this healthy corrected cell, isolate it, and grow it out to have a population of corrected fibroblasts or isogenic controls. And this is a really useful tool to have these healthy isogenic controls because we can see within the same genetic background what the cell looks like if it has the GNE myopathy causing mutation versus what it doesn't. So it's just another one of these cell model systems that will be extremely useful to have for research studies. So in addition to developing those variety of cell model systems, another thing that I've been looking to do in the lab is to develop reliable cell assays that we can test on these cells in the biobank, or also look for any disease biomarkers. So one of the things that I did with the GNE Myopathy Biobank of patient cell lines was perform RNA sequencing to see if we could detect any biomarkers, so any differences between that we could see on an RNA level of the GNE myopathy cells and then the unaffected control cell lines. And what you can see from these plots, looking at the fibroblasts, the myoblasts, or differentiated myotubes of the affected samples versus the unaffected samples, is we weren't seeing any consistent, strong upregulation of any genes at the RNA level. So we weren't able to identify any disease biomarkers from this study, but what we were able to determine at both looking at RNA sequencing and proteomics was we could look at the fibroblast lines versus the myoblast lines. And what we can see is a very strong upregulation of a bunch of genes that are specific to muscles. So we can see all of the ones highlighted in red here, or some of the ones labeled on the right here at the protein level, is that our myoblasts and myotube cell lines were very strongly mimicking muscle cells, which is a very important thing to know, that our myoD conversion, or conversion from the fibroblast skin cells into the myoblast or myotube more muscle-like cells, was occurring very successfully. So in addition to looking at an RNA level, I've also been looking to optimize assays that we can perform on the cells to measure sialic acid production or utilization to get a measure of sort of GNE activity within cell lines. And so there's 3 assays that I'm going to talk about that I've been working with in the cell line. First I'd like to discuss a bit of some of the assays that have been previously used to measure GNE function. And so these are in vitro enzymatic assays, so they're not performed within the cell. And they can be used to measure the GNE enzymatic activity, so either the epimerase or the kinase activity. It's 2 functions. And there are many publications like this that have looked at the GNE protein itself in a cell-free system, looking to see how different mutations in the gene affect the activity levels of the various domains. And there are some pros and cons versus doing these assays in the in vitro system versus an in vivo assay, so within a cell or animal model system. And so in vitro assays are very useful because they're inexpensive generally. They're also non-invasive. They don't require donations from patients. They also have the ability to be very high throughput, so you can test a bunch of different samples very quickly and without too much difficulty. But one of the cons of this is that they occur outside of the biological context. So if you remember from the background I gave you into GNE myopathy, GNE exists within the biosynthesis pathway of sialic acid. So how it interacts within that pathway could have a very important role in how the disease of GNE myopathy occurs. And so this is kind of one of the things that we're looking to overcome doing this in vivo or in a live cell model assays, is that they occur within the biological context. So we can look at how GNE exists within the sialic acid biosynthesis pathway as a whole. The cons of that is that they tend to be more expensive to keep the cells alive. They also require patient tissue donation of some kind, at least for the in vivo assays on cell models, and there can be variation between samples. But given the main pro of it occurring within the biological context, that's why we are super interested in looking at these cell-based assays. So the first one that I'm going to talk to you about is the sialic acid assay, and its sort of overall goal is to determine the overall levels of total and protein-bound sialic acid. So the way that this assay works is we will take the cells, so the wild-type healthy cells, as well as the GNE knockout cells, or eventually cells with mutations in GNE, and we grow them in media that doesn't contain any sialic acid. We can then harvest the membrane proteins of these cell lines, then look to cleave off the sialic acid from these proteins. So looking to remove the sialic acid that exists within these proteins, and then perform a colorimetric assay that can basically measure the levels of sialic acid. So we're looking to measure the levels of sialic acid that exist on the proteins of the cell membrane. And so I have done a few initial experiments looking at these cells using our knockout cell lines that we have in the lab. And so looking at various days of serum starvation, so growing in media free of sialic acid, we can see that there does appear to be a difference in overall sialic acid between our healthy cells and versus the reduced level that we see in the GNE knockout cells. So this requires many more replicates and further experimentation to make to the level of sort of robustness and accuracy that we're looking for, but it's a good initial sign that this could be an assay that we could use to look for differences between the healthy cell lines versus the GNE deficient cell lines. So the second assay that I'm looking to develop is a lectin toxicity assay. So if you happen to catch Dr. Crow's talk, you've heard a lot about lectins and how she's using them as biomarkers. But as a brief refresher, the way lectins work is they bind to sugar chains on the cell surface. So as I mentioned before, sialic acid, which is shown in this sort of pink diamond, is added at the very end of sugar chains on the cell surface. And the lectins bind to these sugar chains. So some bind to sialic acid. So wheat germ agglutinin is one that binds sialic acid, whereas others bind other sugars. So concanavalin A, for instance, binds mannose. And so using these two lectins, we can look to see how strongly they bind to cells based on if they have sialic acid or if they don't have sialic acid. So the way that this assay using these lectins works is we will sort of plate a bunch of healthy or GNE knockout or GNE deficient cells on a plate, and we can treat with various concentrations of these lectins. And these lectins at high enough concentrations are actually very toxic to the cells. So the cells do not like to grow in the presence of high concentrations of these lectins. We leave them growing in these in media with these toxic lectins for several days, and then we can look to measure the amount of cell growth, with the idea being that if the lectin is toxic to the cell, if it's a lectin that binds sialic acid, it will be more toxic to cells with a lot of sialic acid, where if it binds other sugars, it would be more toxic to cells that don't have that sialic acid, that have other sugars as their terminal sugar on the cell surface. So in this way, we can utilize this property of lectins to affect cell growth in cells with a lot of sialic acid versus not a lot of sialic acid. And so in some initial experiments, you can see here, this is the plate of all the cells with the cells stained blue. And as we increase the concentration of the lectins that we're using, that we're treating and growing them with, we see less and less blue cells because more of the cells are not growing or are dying because these lectins are toxic. We can then measure how many cells are left on the plate to see just how toxic these lectins were to the cells. And so looking at here, we can quantify this number and compare how toxic they were to a healthy cell line versus a cell line, a GNE knockout cell line. And what I want you to focus on is that at these high concentrations, we start to see a difference between the wild type healthy cell line in blue versus the knockout cell line in orange. So in this case, when treating with high concentrations of wheat germ agglutinin, which binds to sialic acid, it's much more toxic to the healthy cell lines because they have a lot of sialic acid, which is what we're expecting. Whereas if we treat with concanavalin A, which binds to mannose, it's much more toxic to the GNE knockout cell line because it doesn't have sialic acid, so more mannose is exposed. So this is really another assay that we're super interested in exploring and moving into patient cell lines to see if this sort of trend holds true in our patient cell lines as a way to measure the sialic acid biosynthesis pathway. And then the last assay that I'm working on developing is a PSA NCAM assay. So while the first assay was looking at total sialic acid levels present, the second assay was looking at specifically how sialic acid is used on the sugar chains of the cell surface. This third assay is looking at sialic acid of a very specific protein called NCAM. And this NCAM protein is actually expressed highly in muscle cell lines. And so here's sort of a picture of this protein. And as you can see, all these purple dots represent sialic acid that is attached in these long chains on this protein. So then we can use an assay with antibodies that detect either this protein with all of its sialic acid chains or without all of its sialic acid chains. And then we can look to see if there's more or less of these sialic acid chains present on the proteins of the cell. So this, again, is in the very early stages of developing this assay, mainly just working to optimize the assay, which is a flow cytometry-based assay. But basically, what you can see is that on the y-axis, we can see the NCAM signal, so the protein without the sialic acid. Then on the x-axis, we can see how much of the protein exists with the sialation. So then we can see when we use both of these enzymes together, we can see in this quadrant is all the cells that have the sialated form of this protein, and in this quadrant are all the cells without the sialated form of this protein. So then we can get a percentage of how many of these cells have the sialated form. So in this sample, it happened to be just around 60% of the cells had the sialated form of this protein, whereas 33% had the protein without the sialated form. And we can look in a variety of cell lines to see if this proportion changes. So if we have cells that are deficient in GNE, do we not see as many cells with the polysialated form of this protein? So again, this assay is in early stages of optimizing and working on to get the conditions right, but we hope to eventually be able to use it in our patient cell lines. So that was kind of some of the assays and disease biomarkers that I am looking to optimize. But then sort of the end goal of developing both these cell model systems as well as these assays is to use them to further our understanding of disease mechanism, which can lead to treatments. So that's kind of the end goal in developing all of these preliminary things that I have been. And so one of the things that we're looking to do to understand disease mechanism is first to investigate possible compensatory mechanisms. So other aspects within the sialic acid biosynthesis pathway that could be targeted for potential treatments. One thing to note is that GNE is expressed in all tissues. So this is looking at GNE expression across a wide variety of tissue types. And actually, its expression in skeletal muscle is relatively low compared to all these other tissues. So it's very curious as to why GNE is a muscle-specific disease if GNE is present in all tissues, but the disease is only affecting muscle. And so one thing that we're thinking could play a role is this enzyme called NAGK, which, as you can see from this diagram that I showed you earlier, actually can perform the kinase activity of GNE. So it can perform one of the steps that GNE does within the sialic acid biosynthesis pathway. So it could be used if there's a mutation in GNE in the kinase domain. This could act as a compensatory mechanism to perform the function instead. And interestingly, this protein, NAGK, is actually expressed very low in skeletal muscle. So we're curious to see if that could, if NAGK is playing a compensatory role, so is able to compensate for any mutations in GNE, if the fact that it's very lowly expressed in skeletal muscle could be why it's unable to do this in skeletal muscle. So we're looking to create a variety of knockout cell lines and study this enzyme within the sialic acid biosynthesis pathway as well. And then we're also looking to design and test some GNE expression constructs. So I've created a construct that expresses GNE under the control of a tetracycline response element. And what this means effectively is that we're able to control the level of GNE expression that we're getting from this construct. And so the way this works is we transduce this lentiviral plasmid into a cell line, and then we can treat with a compound or a chemical called doxycycline that induces the GNE to express. So depending on how much of this compound that we use, we can vary the amount of GNE that is expressed. And then we can, measure the sialic acid levels using the variety of assays that I previously talked about to see how expressing GNE affected the sialic acid biosynthesis pathway. And so we can do this mainly to vary the levels of expression to see what level of GNE expression is needed for to correct any defects in sialic acid biosynthesis. And this is a really key thing to know as we're working towards developing treatments, is how much healthy GNE do we need to express in order to see a positive outcome or a positive result in sialic acid production. And then lastly, we can also use these constructs to study pathogenic variants as well as the GNE expression levels. So if we think about GNE function on a range from 0% being lethal, a reduced amount leading to GNE myopathy, a normal amount resulting in healthy individuals, and a very high amount leading to a different disease called sialuria, we can then work to correlate this with not just the GNE function that we're seeing, but how this affects the sialation, both globally, on the cell surface, or on specific proteins, and look to see, how much we need to express GNE to restore sialation from a reduced level that we would see in GNE myopathy individuals to this healthy level. And this would allow us to answer or gain insight into two very, very important questions for therapy or treatment development. So namely, do different pathogenic mutations respond differently to treatments? And then also what functional GNE expression levels are needed to restore proper sialation. And both of these things would be really crucial things as we move towards developing a treatment or therapy. So just some concluding key takeaways that I hope you've gotten from my talk is that cell model systems are an extremely useful tool for GNE myopathy, both research and therapy development. Also, the development of robust in vivo assays that can measure sialic acid biosynthesis will eventually lead to us being able to use them to determine the efficacy of potential therapies. So using them to measure how effective a treatment is at restoring proper sialic acid biosynthesis. And then lastly is that additional research into GNE expression levels and pathogenic variants can also provide insights into best therapeutic strategies. So with that, I have many thanks to give first to all the members of the LEC Lab who have been helping me on this project and providing insight and guidance, as well as my thesis committee, who has been also giving me advice throughout this project. Also, all of our collaborators that I've gotten help with on this project, as well as the NDF for not only providing some of the resources for me to be able to do my science, but also the opportunity to speak today. And lastly, a huge thank you to the patients, because without their generous donation, much of the work that I've done would not be possible. So thank you.
Great. Thank you so much, Katherine, for sharing some of the interesting research you're undertaking in the lab. It's interesting, but I'm a little bit biased. It's definitely quite difficult to give a talk to a mixed audience of scientists and non-scientists, and I think you struck a right balance for both. And hopefully everyone listening today can take away something from the talk. So I guess I can kick off with one question. I think some non-scientists in the audience may be wondering if you can explain the advantages of using a knockout cell line and why you had to create that over, say, patient cell lines that you corrected. Can you give some insight into that?
Yes. So as I touched on briefly, the patient cell lines or the patient mutations that we see are not complete loss of GNE activity. So we see a reduction in enzymatic activity, whereas in a knockout cell line, we have a complete loss of GNE function. So both are super useful tools, whereas the patient mutations will give us an intermediate result from healthy. If we're looking at GNE activity levels, a complete knockout will, in theory, give us no GNE expression or activity levels. So it's a good control to have where we have a healthy cells that are completely healthy, that have normal GNE expression, a knockout that has completely zero GNE expression, and the patient mutations will be usually somewhere in the middle in regards to their GNE enzymatic activity.
Correct. Yeah, that's something we in the science world called like a negative control, where we'll get, if we have no GNE at all, the reading for whatever assay we're trying to calibrate will be null or zero, as opposed to normal, and then patient cells lie somewhere in the middle of the spectrum. So it's a really useful tool to calibrate some of the cellular assays that Katherine spoke about today. So that brings me to my next point. So you touched upon the functional assays in your talk, for example, measuring sialic acid, lectin toxicity assays. Can you expand a bit upon why they are important in thinking about therapies and clinical trials?
Mm-hmm. Yes, so one really important thing that when we're developing therapies and clinical trials is we need a way to measure if the therapy or treatment that we're providing is working. And so you need a way of measuring what a healthy patient or cell looks like versus a patient with GNE myopathy. You need a way of something to measure. So when you perform the treatment, you can then look to see if the GNE myopathy patient, the levels of that assay are recurring or changing to a more healthy state. And so having that clinical outcome measure of something to determine if the treatment is working is super important, and something that would be required for any sort of clinical trial is having something to measure to show the treatment's working.
And these assays you're developing are presumably agnostic to the therapy. So they would be applicable to cells treated with a gene therapy construct, or also like MANAC. So these are sort of functional measures that then scientists can see if they've been restored, a biological function in GNE cells that have been restored, to know if a therapy is working or not working. That's why these assays are very important in thinking about testing and also going towards clinical trials. Because, I've stated in previous talks, GNE is a slowly progressing disease. So it's really difficult to know if you give a therapy to a patient, if it's worked or not. And unfortunately, simply asking a patient, hey, did this therapy work? That's not considered very scientific. So we need sort of things to be able to measure that's occurring on a molecular level to know if a therapy has made a positive impact or not. So there is a question here in the Q&A box. by Tara. So let me read this out. This is a question from one of our patients who could not attend today. Will this study be used for treatment? Can you please explain in a form for patients to understand? It's a very broad question, but if you want to take a stab at...
So most of the work that I presented today was working to develop either assays that would be used in clinical trials or that could be used as research tools when people go to test various treatments. So not really developing the treatments themselves, but providing many of the tools that both researchers and clinicians for eventual clinical trials will need to be able to test therapies or measure their effectiveness.
Yeah. So usually before going to clinical trials for rare diseases, there needs to be a sort of framework set up to better understand the disease, having a disease model, and also some of these molecular assays that Katherine spoke about. Usually patient foundations will invest resources in order to develop these really critical building blocks before we can get to clinical trials. So that's I hope that answers Tara's question, but please expand upon your question if you don't think we answered that. And let me think, is there any other questions? I can ask you another one. So you spoke about animal models and previous attempts at generating GNE models, none of them having sort of the ideal characteristics. What do you think are the set of characteristics then that would make for a really good model, you think, of GNE myopathy in mouse?
Mm-hmm. Well, I mean, the main thing that has been a challenge to get is a phenotype that mimics the phenotype of that seen in GNE myopathy patients, so namely the progressive muscle loss. Because given that that would be the thing that we would be looking to correct in humans, it would be very useful to have that in a mouse, also on a sort of reliable time frame where you could, do experiments relatively quickly. So those would be the main thing.
Yeah, and ideally something that is that mirrors maybe the late onset nature, so not a congenital not congenital phenotypes, and maybe so the muscle specificity, I guess, is important, and the late onset, and then something that can be linked to decreases in sialic acid biosynthesis, maybe, I guess, on a molecular level that can then be measured. But we may be asking for too much, considering that they have that additional gene. You spoke about the CMAH, and they have the two forms of sialic acid. So that might not completely be, we might not be able to completely model that disease in a mouse. So in a way that faithfully recapitulates the human condition. So that's something we have to definitely think about. Oh, Marco has a question. Wait, one more. There are other genes involved in the sialic acid biosynthesis pathway. Do they cause disease when mutated? Do any of them also cause a muscle disease? If not, does that imply that there are other functions for the GNE beyond biosynthesis pathway? That's a bit of a, that's a lot to think about.
Yes, long story short, there are other genes involved, and so the one of them that I touched on, the CMAH, there has been studies that show, at least in a mouse model, that when mutated, it can impact the severity of muscular dystrophies. But given the differences between the sialic acid biosynthesis pathway in humans compared to other species, especially non-mammals, it's a challenging thing to investigate, but yeah, it is definitely something to look into. But as of now, our leading hypothesis is that GNE's sort of main function is its role in the sialic acid biosynthesis pathway.
Okay, thank you for that. And feel free to email me or Katherine directly if you have any further questions. And yeah, and this talk will be made available shortly yeah, through the NDF website. And I believe next week is the final week of this summer's webinar series. So thank you, everyone, and good job, Catherine. Have a good weekend, everyone.